Stomata in Greek means mouth and its singular is stoma. These are structural features of aerial organs like leaf in most plants. The term stoma represents microscopic pore or hole through which plants breathe. These tiny openings on the surface of the leaves are called stomata, which allow communication between the interior and the external environments. The stomata is guarded by the pair of specialized cells called as guard cells, which responds to environmental signals by changing its dimensions and thus regulates the size of the pore or stomatal aperture. Air enters the plant through these openings and contains carbon dioxide and oxygen, which are used in the processes of photosynthesis and respiration, respectively. Oxygen produced as a byproduct of photosynthesis diffuses out to the atmosphere through these same openings. Also, water vapor is released into the atmosphere through these pores in a process called transpiration. Nearly 120 years ago, Francis Darwin in 1898 showed that stomata on leaves respond to environmental stimuli. The guard cells are responsible for regulating the opening and closing of stomata. Stomata covers 1 to 2% of leaf area. It is minute pore present in soft aerial parts of the plant. Algae, fungi and submerged plants do not possess stomata. Distribution of stomata. Distribution of stomata varies between monocot and dicot plant species. Stomata are present in the sporophyte generation of all land plant groups except liverworts. Dicotyledons usually have more stomata on the lower epidermis than the upper epidermis. Monocotyledons, on the other hand, usually have the same number of stomata on the two epidermises. In plants with floating leaves, stomata may be found only on the upper epidermis and submerged leaves may lack stomata entirely. Size of stomata varies across species with end-to-end lengths ranging from 10 to 80 micrometer and width ranging from a few to 50 micrometer. It also varies between the underside and top side of the leaves. Stomata are found more on plant leaf surfaces under high light, lower atmospheric carbon dioxide concentrations and in moist environments. Structure of stomata. The stomata are minute pores which occur on epidermal surface of the leaves and also some herbaceous stems. Each stoma remains surrounded by two specialized epidermal cells called guard cells. These guard cells are also surrounded by other specialized epidermal cells called subsidiary cells or accessory cells. The stomata may occur on any part of a plant except the roots. The outer wall of the guard cells is thin and elastic, while inner wall is thick and non-elastic. Guard cells are epidermal cells, but due to presence of chloroplast, they are different from that of epidermal cells. Usually in the leaves of dicot plants, the stomata remain scattered, whereas in monocot plants, they are arranged in parallel rows. The structure of guard cells in monocots is dumbbell shaped, while in dicot it is kidneys or bean shaped. Each guard cell is a modified epidermal cell showing a prominent nucleus, cytoplasm and plastids. The wall of the guard cell is differentially thickened. The inner wall of each guard cell facing the stoma is concave and is thick and rigid. The outer wall is convex and is thin and elastic. The gas exchange between plants and the environment depends on the guard cells. The guard cells from the lower surface of the leaf are more sensitive to the environmental factors and provide the major part of the leaf gas exchange. The guard cells are produced in pairs with a gap between them that forms a stomatal pore. The stomatal pores are largest when water is freely available and the guard cells are turgid, and closed when water availability is critically low and the guard cells become flaccid. The guard cells are surrounded by a variable number of epidermal cells called subsidiary cells. The osmotic pressure of the stomata is far larger in the guard cells than in the subsidiary cells. This ratio shifts in favor of the subsidiary cells when the stomata are closed. Subsidiary cells provide a cushion for the adjacent epidermal cells, while guard cells expand or contract, thus they support in the movement of guard cells. The function of guard cells is much determined by their structural features. Each guard cell has a cytoplasmic lining and a central vacuole. Its cytoplasm contains single nucleus and a number of chloroplasts. The chloroplasts of guard cells are capable of very poor photosynthesis because of the absence of enzyme rubisco. The size and shape of stoma and guard cell varies from plant to plant. When fully open, the stomatal pore measures about 3 to 12 micrometers in width and 10 to 40 micrometers in length. In many gymnosperms and xerophytic plants, that is, those plants growing in desert, the stomata are present embedded deeply in the leaves so that they are not exposed to sunlight directly. Such deeply embedded stomata are called sunken stomata. This is an adaptation to check excessive transpiration in these plants. Stomatal index. Stomatal index is the percentage which the number of stomata forms to the total number of epidermal cells, each stomata being counted as one cell. Stomatal index can be calculated by using the following equation, where SI is the stomatal index, S is the number of stomata per unit area, and E is the number of epidermal cells in the same unit area. Number of stomata or stomatal frequency. The number of stomata in a definite area of leaf varies from plant to plant. Xerophytes possess larger number of stomata than mesophytes. Number of stomata per square centimeter is 1000 to 60,000 in different plant species. The number of stomata per unit area of leaf is called stomatal frequency. Stomatal frequency of trees and shrubs is higher than herbs. For example, the number of stomata in the leaves of tomato may range from 1200, while in scarlet oak it may be more than 1 lakh. Stomata nearly occupy 1 to 2% of the total leaf area when fully open. In isobilateral leaves, that is in monocots, approximately the same number of stomata are found on upper surface, that is adaxial surface, and lower surface, that is the abaxial surface. But in dorsiventral leaves, as in case of dicots, the number of stomata on the upper surface is much less in comparison to those found on the lower surface. Distribution and types of stomata. Depending upon the distribution and arrangement of stomata in the leaves, 5 categories of stomatal distribution have been recognized in plants. First is apple or mulberry type or hypostomatic type. Here, the stomata are found distributed only on the lower surface of the leaves, and examples are apple and peach. Potato type. Stomata are found distributed more on the lower surface and less on its upper surface, for example, in case of potato, cabbage, beans, etc. The third type is oat type or amphistomatic type. Here, the stomata are found distributed equally upon the two surfaces, and the examples are maize, oats, grasses, etc. The fourth type is water lily type or epistomatic type. Here, the stomata are found distributed only on the upper surface of the leaf, and the examples are water lily, nymphea, and many aquatic plants. The fifth type is Potamogeton type or astomatic type. Stomata are altogether absent, or if present, they are vestigial. For example, in Potamogeton and submerged aquatics. Four types of stomata have been recognized on the basis of their structure. Anomacytic type. In these stomata, accessory cells are absent. The guard cells are surrounded by ordinary epidermal cells, for example, in families of Ranunculaceae, Cucurbitaceae, Papavervaceae, and Malvaceae. Anisocytic type. In these stomata, the guard cells are surrounded by three accessory cells. Of these, two are larger, whereas one is smaller in size, and the example is family Brassicaceae. Diacytic type. In these stomata, the guard cells are surrounded by two accessory cells. Their common walls are at right angle to the walls of the guard cells, and such stomata are found in the family Caryophyllaceae and Acanthaceae. Paracytic type. In these stomata, the guard cells are also surrounded by two accessory cells, but their common walls are parallel to guard cells, and the examples are seen in the families Rubiaceae and Fabaceae, etc. On the basis of development, there are three types of stomata. Mesoperigynous type. In this type of stomata, guard cells as well as subsidiary or accessory cells both are developed from one mother cell. Examples are in Rubiaceae and Brassicaceae family. Perigynous type. In this type, guard cells are formed from mother cells while subsidiary cells from nearby mother cells, and examples are seen in Cucurbitaceae family. Mesoperigynous type. In this type, guard cells and one subsidiary cells is formed from mother cell while other subsidiary cells develop independently. Example is seen in Ranunculaceae and Caryophyllaceae family. Graminaceous. The graminaceous stoma possess guard cells of which the middle portions are much more narrower than the ends, so that the cells appear in surface view like dumbbells. They are commonly found in Graminae and Cyperaceae families of monocotyledons. Coniferous stomata. They are sunken and appear as though suspended from the subsidiary cells arching over them. In their medium parts, the guard cells are elliptical in section and have narrow lumina. At their ends, they have wider lumina and are triangular in section. The characteristics of these guard cells is that their walls and those of the subsidiary cells are partly lignified and partly non-lignified. Functions of stomata. Carbon dioxide gain and water loss. Carbon dioxide, a key reactant in photosynthesis, is present in the atmosphere at a concentration of about 390 ppm. Most plants require the stomata to be open during daytime. The air spaces in the leaf are saturated with water vapor, which exits the leaf through the stomata, and this is known as transpiration. Therefore, plants cannot gain carbon dioxide without simultaneously losing water vapor. Alternative approaches. Ordinarily, carbon dioxide is fixed to ribulose bisphosphate, that is RUBP, by the enzyme Rubisco in mesophyll cells exposed directly to the air spaces inside the leaf. For several reasons, Rubisco needs high carbon dioxide concentrations, which means wide stomatal apertures and, as a consequence, high water loss. Narrower stomatal apertures can be used in conjunction with an intermediary molecule with a high carbon dioxide affinity. This is PEPcase or Phosphoenolpyruvate carboxylase. Retrieving the products of carbon fixation from PEPcase is an energy intensive process. However, as a result, the PEPcase alternative is preferable only where water is limiting, but light is plentiful and temperature is high. CAM plants, C3 and C4 plants. Stomata stay open all day and close at night. CAM plants' stomata open during the morning and close slightly at noon and then open again in the morning. A group of mostly desert plants called CAM plants by the name Crassulacean acid metabolism open their stomata at night when water evaporates more slowly from the leaves and use PEP carboxylase to fix carbon dioxide and store the products in large vacuoles. The following day, they close their stomata and release the carbon dioxide fixed in the previous night into the presence of Rubisco. This saturates Rubisco with carbon dioxide, allowing minimal photorespiration. This approach, however, is severely limited by the capacity to store fixed carbon into the vacuoles, so it is preferable only when water is severely limiting. Opening and closure of stomata. Most plants do not have the aforementioned facility and must therefore open and close their stomata during the daytime in response to changing conditions such as light intensity, humidity and carbon dioxide concentration. It is not entirely certain how these responses work. However, the basic mechanism involves regulation of osmotic pressure. When conditions are conducive to stomatal opening, that is, high light intensity and high humidity, a proton pump drives protons from the guard cells. This means that the cell's electrical potential becomes increasingly negative. The negative potential opens potassium voltage gated channels and so an uptake of potassium ions occurs. To maintain this internal negative voltage so that entry of potassium ions does not stop, negative ion balance the influx of potassium. In some cases, chloride ions enter, while in other plants the organic ions like malate is produced in the guard cells. This increase in solute concentration lowers the water potential inside the cell, which results in the diffusion of water into the cell through osmosis. This increases the cell's volume and turgor pressure. Then, because of rings of cellulose microfibrils that prevent the width of the guard cells from swelling and thus only allow the extra turgor pressure to elongate the guard cells, whose ends are firmly held in place by surrounding epidermal cells, the two guard cells lengthen by bowing apart from one another, creating an open pore through which gas can move. When the roots begin to sense a water shortage in the soil, abscisic acid or ABA is released. ABA binds to receptor proteins in the guard cell's plasma membrane and cytosol, which first raises the pH of the cytosol of the cells and cause the concentration of free calcium to increase in the cytosol due to influx from outside the cell, and release the calcium from internal stores such as the endoplasmic reticulum and vacuoles. This causes the chloride and inorganic ions to exit the cells. Secondly, this stops the uptake of any further potassium into the cells and subsequently the loss of potassium. The loss of these solutes causes an increase in water potential, which results in the diffusion of water back out of the cell by osmosis. This makes the cell plasmolized, which results in the closing of the stomatal pores. Guard cells have more chloroplasts than the other epidermal cells from which guard cells are derived. Their function is controversial. Conclusion: the stomata plays vital role in exchange of gases and moisture from internal environment of the cell to the exterior. In majority of plants, the stomata open and close during day and night time respectively. The opening and closing of stomata is stimulated by water, potassium ion, light intensity and carbon dioxide concentrations in the guard cells. Open stomata enable the uptake of carbon dioxide for photosynthesis, while closure prevents excessive loss of water during drought conditions.